warm welcome to all the students. Today, my class will discuss on Mycobacterium tuberculosis, and that too on the genetics of a reference strain. So before starting today's lecture, I would like to discuss some of the important points about the Mycobacterium tuberculosis. So basically, it is a bacterium. Uh, gram, we can say that it is neither a gram positive. Strictly, if we say that it is neither a gram positive nor a gram-negative bacterium. It is an aerobic bacteria, then non-sporing, non-motile, and non-capsulated. And the term mycobacterium basically comes from the term mycology, that is that it looks like fungus-like bacteria. That is, it shows filamentous-like growth, just like mycelium on a solid media, and it also on the liquid media, the characteristics shown by the bacterium is fungus-like growth. So regarding its history, so the first mycobacterium was discovered by Hansen in 1868. That was the Mycobacterium leprae. So it is basically divided into three parts or three types. That is the Mycobacterium tuberculosis leprae and non-tubercle Mycobacterium. So Mycobacterium tuberculosis, before getting discovered, the first Mycobacterium was leprae discovered by Hansen in 1868. And Mycobacterium tuberculosis was found by Robert Koch in the year 1882. Some of the important characteristics of Mycobacterium tuberculosis is that it causes pulmonary tuberculosis and spreads through droplet infection, mostly while coughing, sneezing, and talking. And it shows resistance to decolorization even with strong acids. So that's why the term was given to it was acid fast bacilli, about its characteristics of resisting decolorization by acid. Therefore, these are some of the important characteristics I have discussed with you. And basically, it is mostly survives within the macrophages. And it have a unique mechanism which helps it in a latent stages and mostly start dividing multiplications and symptoms when the patient's immune system is lowered. WHO have declared it one of the most globally one of the important diseases responsible for morbidity and mortality throughout the world, especially in the developing or underdeveloped countries where malnutrition is common, and it has to be addressed very effectively and efficiently so that it should be properly eliminated. So here we will discuss about the genomics of the Mycobacterium tuberculosis. So first of all, I would like to discuss about the term, that is the H37RV. So what is H37RV? So it was first discovered by Stewart Cole and his colleagues in the year 1988, and it gave TB research a much needed boost by clarifying the pathogenic genetic makeup and identifying numerous novel gene products for mechanistic and structural characterization, as well as possible new therapeutic targets. See, one thing I would like to discuss with you, that if the basically any bacterium, either it can be treated by its phenotype, that is on the basis of its appearance, structure, general routine characteristics like growth characteristics, then staining characteristics, then which type of bacterium it is, or which type of physical characteristics it shows. And on that, the medication is given. But in genotype, when we go through the genotype, then we get a lot more clarity on the genetic makeup of any bacterium or pathogen, and it becomes quite easy for us to identify those genes which are responsible for causing pathogenesis in the host. And when we identify those genes, it becomes quite easier for us to, in the help of to research on them and to find the targets of these genes. So here, the H37RV discovery was many was one of the most revolutionary discovery in the field of diagnosis of tuberculosis, which it because it gave the entire genomic structure of the mycobacterium, which not only helped us, or which not only helped the scientist to study the various characteristics on the genetic level, because all the informations regarding the any of the hosts, any of the living beings is coded on the gene. And if we decipher it, if we decode it, then it becomes quite easy for us to understand all those structures or all those characteristics, and we can treat it on the genetic level. And it becomes quite effective in comparison to some of the conventional methods. So here, what the Stuart Cole and colleagues saw when they discovered or isolated the H37RV gene, that the genome comprises of 44 lakhs 11,529 base pairs and has an average G plus C content of 65.6%, with some areas with an exceptionally high G plus C content, that is greater than 80%, were detected and found to correspond to a novel gene family. So it was quite because usually we see that if you would be knowing something about the base pairing or nucleotides, you'd be knowing that purines and pyrimidines are there, adenine, guanine, and cytosine and thymine. So basically, purine makes base pair with pyrimidine. So here basically saw that guanine and G plus C content was more than 60%. It was almost 65.6%, which is quite unusual in comparison to other where adenine thymine is also found in more amount, adenine thymine pairing. Okay, so here we saw that almost were detected and found to correspond to a novel gene family by means of bioinformatics. Bioinformatics is a science which deals of deciphering the which deals with the study of pathogens or any living beings with the help of some of the softwares or computer softwares where genes are studied. So by means of bioinformatics, 50 genes encoding stable RNA species and 3,924 genes encoding proteins were identified, and these account for greater than 91% of the potential coding capacity. H37RB is the standard laboratory reference strain of Mycobacterium tuberculosis, the bacterium causing tuberculosis. This was first isolated in 1905, and the name signifies its history. H represents human origin. 37 indicates it was the 37th strain studied at Trudeau Sanatorium R denotes rough colony morphology, and V stands for virulent. Next, what is H37RB? So the strain H37 was originally, it was first isolated. See here, one thing I would like to remind you that this genome was sequenced. See that is the difference, that Stewart, Cole and colleagues have successfully sequenced the genome of H37RB, but the reference strain from the bacterium from which H37RB was taken or sequenced was first isolated in 1905. So dating back to the history, so the strain H37 was originally isolated in 1905 and gained attention for its noted virulence in the guinea pig model, a distinctive characteristics used in the classification of human tuberculosis. In the early 1900s, which was originally isolated from a 19-year-old patient with chronic pulmonary tuberculosis by Edward R. Baldwin in 1905. In order to obtain stable avirulent derivatives of H37, in 1935, William Steenken carried out a dissociation study based on aging of H37 bacilli on solid egg media. So here I would like to explain that the first isolation was of H37 was in 1905, which was a virulent strain, and it was found by Edward R. Baldwin in the year 1905. And in the year 1935, which was a virulent strain, means that it has the capacity to cause infection or the invade or cause damage to the host tissues. And if any, but for the study of the bacteria, it was used as a reference strain, means if any of the Mycobacterium was isolated, it was just like a control or reference bacteria to study the other Mycobacterium. So for the study, it was made evirulent by repeated cultures on a solid egg media. And the parental virulent H37 was inoculated onto solid egg media at pH 6.2. The resulting culture was allowed to age for 3 to 4 months at 37 degrees centigrade. By the end of the extended incubation, the original dry, discrete colonies lysed and transformed into a confluent viscous mass. In the midst of the viscous mass, secondary growth with different colony morphology emerged. The new growth, when picked and cultured on fresh media, produced no disease in guinea pigs and was designated as 37RA, A for avirulent. So the virulent counterpart, that was a rough colony morphology, was named H37RV, and the original H37 was discontinued. H37RA and its virulent counterpart H37RB has been widely used as a reference strains for studying virulence and pathogenesis of Mycobacterium tuberculosis since 1940s. And the H37RA also used as an adjuvant to boost immunogenicity during immunization. The Mycobacterium tuberculosis strains H37RB and H37RA, the most commonly used controls for Mycobacterium tuberculosis identification in the clinical research laboratory settings. So this is a brief history of the H37RB, which was used as a reference strain to study the pathogenesis as well as inducing immunogenicity in the individuals. So coming to the next slide. So Mycobacterium tuberculosis H37RB is a virulent laboratory adapted strain classified as a gram-positive actinobacterium. specifically acid-fast bacilli. It is the primary reference strain for Mycobacterium tuberculosis research, featuring a genome size of approximately 4.4 million base pairs and a complex cell wall structure rich in lipids. Key structural and classification aspects. So strain designation, H37RV stands for human 37, isolated in 1905, and RV for rough virulent. Cell envelope features a unique waxy cell, acid fast, containing high concentrations of mycolic acids, lipids, and glycolipids that contribute to its pathogenicity. Genome structure. The genome is a single circular chromosome. It is characterized by high GC content and the presence of specialized gene families such as PGRS family, 63 members, which are involved in host interaction and pathogenicity. Molecular features. Proteomic analysis often categorize its structural proteins into functional groups like cell wall, biosynthesis, intermediary metabolism, and virulence factors. Taxonomy classified with the order of actinomycetes, family Mycobacteriaceae, genus Mycobacterium. Mycobacterium tuberculosis H37RB is used globally as the benchmark for studying tuberculosis pathogenesis, drug resistance, and vaccine development. Now here we know that it is one of the most global pandemic or global, we can say, one of the most prevalent mycobacterium, which have led to quite large number of deaths throughout the world, and it's a chronic disease. And the complete genome sequence is best characterized strain of Mycobacterium tuberculosis H37RB has been determined and analyzed in order to improve our understanding of the biology of this slow-growing pathogen and to help the conception of new prophylactic and therapeutic interventions. The genome comprises, as we have already discussed, that it is of 44 lakhs 11,529 base pairs, contains around 4,000 genes, and has a very high guanine cytosine content that is reflected in the biased amino acid content of the protein. Mycobacterium tuberculosis differs radically from other bacteria in that a very large portion of its coding capacity is devoted to the production of enzymes involved in lipogenesis and lipolysis, and to two new families of glycine-rich proteins with a repetitive structure that may represent a source of antigenic variations. In 1993, the gravity of the situation led to the World Health Organization to declare tuberculosis a global emergency in an attempt to heighten public and political awareness. Radical measures are needed now to prevent the grim prediction of the WHO becoming reality. The combination of genomics and bioinformatics has the potential to generate the information and knowledge that will enable the conception and development of new therapies and interventions needed to treat this airborne disease and to elucidate the unusual biology of its etiological agent, Mycobacterium tuberculosis. The characteristics feature of the tubercle bacilli included slow growth, dormancy, complex cell envelope, intracellular pathogenesis, and genetic homogeneity. So aiming at a better now for better understanding of the virulence and immunity in MTB, the complete genome of a strain H37RB has been sequenced. Among the approximate 4,000 genes in the MTB genome, nearly 25% of them are annotated as hypothetical proteins. So 25% have been, after studying them, has been termed as hypothetical proteins, which are encoded by predicted open reading frames but do not have any confirmed functions. In many species, hypothetical proteins can play important roles in the survival of pathogens and the progression of associated infectious diseases. In MTB, some of these hypothetical proteins have been experimentally characterized. For example, RV0079, which was found to be a DOS-R regulon playing an inhibitory role in protein synthesis and interacting with toll-like receptors to promote cytokine secretion. See, basically what happens that if in simple terms, if like to make you understand that mostly the immunity which develops during the infection is the cell mediated immunity, T cell dependent immunity and antibodies do not have any role in the immunity against the Mycobacterium tuberculosis. So in that cases, it is the role of cytokines like interferon, tumor necrosis factors are mostly they are prevalent. So they are come under the cytokine, and here there is that what happens that these regulons inhibit these protein synthesis and interacting with TOL to promote cytokine secretion. These previous results indicate that HAP could now also play important roles in Mycobacterium tuberculosis, thus enhancing the growth and multiplication of tuberculosis. Okay. And however, the functions of most hypothetical proteins in MTB are still unclear. Bioinformatics has revealed that many hypothetical proteins are actually enzymes, kinases, or binding proteins with over 50% annotation possible through homology modeling, aiding in the discovery of new drug targets, that is RV0817 and RV2927. So if we know about the original loci of this gene, we can easily target or synthesize some drugs which can easily target on the genetic basis of this mycobacterium. And certainly, if we target, then these genes would not be able to secrete those enzymes which helps or initiate the pathogenesis of Mycobacterium. Certain hypothetical proteins, including those in the P or PP family, are implicated in immune evasion, intracellular survival, and metabolic adaptation. Some HPs are overexpressed in drug-resistant strains, suggesting that they may neutralize antibiotic effects. So here, in addition to hypothetical proteins, which is often labeled as HP or hypothetical protein, Mycobacterium tuberculosis H37RB secretes a wide variety of proteins through its SEC, TAT, and type 7 ESX systems. These six secreted proteins are crucial for virulence, immune evasion, and nutrient acquisition. Major non-hypothetical proteins secreted by H37RB include ESAT6, that is ESXA, and RB3875, a 6 kilodalton early secretory antigenic target central to pathogenesis, often secreted with CFP10. CFP10, that is the ESX uh, and ESXB and RB3874, a 10 kilodalton is culture filtrate protein that forms a 1:1 heterodimeric complex with ESAT6. So ESAT6 and CFP10, two genes which interact together, and both of them enhances the pathogenesis of Mycobacterium. Other are AG85 complex, which include AG85A, B, and C. AG antigen 85 proteins uh, are mycolyl transferase involved in cell wall synthesis and strongly stimulate T cell responses. Then MPT64 is there, which is a major secreted protein frequently used as a diagnostic antigen. It can impair the host macrophages unfolded protein response. Then MPT53 is there. Then MPT32, and these are some of the major gene which secrete some of the important proteins, virulence factors, and antigens. In addition to that, there is other family proteins, that is the P and PPP family proteins, which are rich in proline glutamic acid or proline glutamic acid, and frequently exported by the ESX5 system to modulate host immunity, in which includes the proteins RPP18, which is by genes RB1196. So it is secreted by the gene RB1196, which is the component of vaccine candidates. Then PP2, which is secreted by RB256C, translocate to the macrophage nucleus to suppress INOS expression. Then PE or PPE proteins, various members are involved in nutrient acquisition and immune evasion. Third is the ESX1 and other secretion system associated proteins, that is the ESP. So here some of the secretion system associated proteins are ESPA, ESPC, and ESB-B. And next is the ESX-U, ESX-J, and ESX-N. So enzymes and stress response proteins, others are some of the important proteins which is secreted are the PKNG, PKNF, and PKNA secreted or cell surface protein kinases that manipulate host signaling such as inhibiting phagosome, lysosome fusions, PTPA, then which is secreted by gene RB2232, protein tyrosine phosphatase that cause poor phagosomal acidification, then SAPM RB3310, a phosphatase that inhibits phagosome maturation, then ZMP1 are secreted by RB0198C gene, aging metalloprotease that prevents inflammasome activation. Then GRO-EL1 and GRO-EL2 that they are chaperones found in the extracellular environment involved in stress response, and LUCA secreted by RB3723 required for the import of fatty acids and cholesterol inside macrophages. So these are some of the important proteins and enzymes which are secreted by the genomes of Mycobacterium tuberculosis H37RB. Now, if we summarize it, we can see here that if in the tabular form, that ESAT6 protein name is the ESAT6 secreted by the gene RB3875, and it is responsible for virulence and membrane damage of the host. Then CFP10 RV3874 complex with ESAT6 immune modulation, antigen 85 complex is responsible for mycolide transferase, cell wall, and antigen. MPT64 antigen inhibits host protein response. PTPA inhibits phagosome lysosome fusions. ZMP1 inhibits inflammasome activation. PPE2 suppress inos. CPNT NAD plus hydrolysis activates necroptosis. So H37RB is the standard drug susceptible reference strain of Mycobacterium tuberculosis used to identify MDR-TB mutations. So here, this was it is a drug sensitive strain. It is not a drug resistant strain, but it can be used even as a reference strain to identify MDR-TB because most of the drug resistance is caused by the mutation in the susceptible bacteria. As we all know that there are different type of mutations which helps the bacterium to develop resistance against some of the important drugs. So here H37RB can also be used to study, or it can be used as a reference genome to study multi-drug resistant tuberculosis. So MDR-TB strains are identified by comparing their genetic sequences, and some of the important genes which are responsible for multi-drug resistance are the RPOB, CATG, and INHA to the H37RB genome to find mutation causing resistance, often revealing differently expressed genes. Key connections between H37 and H37RB and MDR-TB are reference genome. Researchers compare MDR-TB clinical strains against the H37RB genome to identify new resistance-associated genes. Differential expression studies have identified unique genes, MTB192 expressed in MDR-TB strains that are absent or different in the standard H37RB strain. Metabolic difference compared to H37RB, MDR-TB strains show specific alterations in metabolic pathways, including fatty acid and arginine biosynthesis. Virulence study. While H37RB is highly virulent, some MDR-TB strains show different, sometimes attenuated virulence profiles in comparative studies. So the comparison helps in understanding the molecular mechanism behind the development of multi-drug resistance. Mycobacterium tuberculosis MTB strain H37RB is the standard fully virulent drug susceptible laboratory reference strain used to study TB and develop a new drug. It is not inherently resistant, but it serves as a benchmark against which drug resistant strains are compared, as resistance developed through genetic mutations such as SNPs and deletions, which are then analyzed using H37RB as the wild type genetic reference. Key aspects of H37RB and drug resistance include reference genome and drug target discovery. The H37RB genome is used to identify essential non-homologous enzymes that act as drug targets. Studies identify hundreds of essential proteins for new effective therapeutics. Genetic basis of resistance will H37, while H37RB is susceptible, in vitro studies used to generate resistant mutants, example via isoniazid exposure to identify mechanism like mutations in CATG for isoniazid or RPOB for rifampicin. Comparative studies H37RB is compared with MDR strains to identify regulatory networks. Example is 3-2 component system and the CRP regulator that mediate resistance, particularly those affecting envelope components and drug efflux. Virulence study, while H37RB is virulent, it can undergo in vitro attenuation, but it remains the key strain for understanding how NTB evolves in response to therapeutic pressure. So here we can see that what is the how do we compare and analyze the H37RB with the MDR-TB, and thus H37RB is the susceptible baseline, not the source of resistance itself. It is critical for and useful in studying the map and genetic pathways and mechanism by which Mycobacterium tuberculosis becomes resistant to drugs. So it also helps that whatever there is any alteration in metabolic pathways, so that it escapes the mechanism of drug effect of drug, or how it neutralizes the effect of drug by altering the structure or altering its metabolic functions. So all those factors can be easily understood by analyzing the H37RB as well as the MDR-TB mutant strains. So reference resistance in Mycobacterium tuberculosis primarily driven by chromosomal mutations in specific genes that alter drug targets or activate efflux mechanism. Key genes include RPOB for rifampicin, CATG and INHA, isoniazid, GYRA, fluoroquinolones and EMB-B ethambutol. Rifampicin resistance over 90% of cases are caused by mutations in the 81 base pair hotspot region of the RPOB gene, which encodes the subunit of RNA polymerase. So here, one of the most important is the rifampicin resistance. Second is the isoniazid, primarily caused by mutation in the CATG gene, catalase peroxidase, specifically codon 315 of the INHA promoter region. Then comes fluoroquinolone resistance, mainly due to mutations in the quinolone resistance determining region, QRDR of the GYRA gene. Ethambutol resistance, frequently associated with mutations in the EMB-B gene, which involved in cell wall synthesis. Streptomycin resistance associated with mutations in RPSL, that is the ribosomal protein S12 and RRS16S ribosomal RNA genes. Intrinsic and general resistance, the YB7 gene acts as a regulator for intrinsic resistance to multiple antibiotics. Additionally, the MMR gene mediates resistance through active drug excretions. These genetic markers are used in rapid molecular diagnostics to detect multi-drug resistant TB. So all these can be easily, so on the genetic study, we can easily recognize which type of resistance is there in the, against which type of drugs the mycobacterium is showing resistance, and we can easily identify those target genes. Now, next, come to the, hence the genetic mutation in the INHA or its promoter region causes the overexpression or mutation of the INHA, which can lead to cross resistance to isoniazid and prothionamide. The frequency of cross-resistance mutations in INHA and or its promoter region varied in different geographic regions, ranging between 13.8% to 100%. The CAT gene codes the catalase peroxidase enzyme in Mycobacterium tuberculosis, which is essential for activating the prodrug isoniazid into its bacterial form. Mutations in this gene, particularly at codon 315, are the primary mechanism for high-level isoniazid resistance, as the altered enzyme fails to convert the drug efficiently. Key aspects of CATG gene is that the CATG enzyme is a multifunctional enzyme that protects the bacteria against reactive oxygen species and acts as a peroxidase. Mechanism of resistance: when the CAT gene is mutated, the catalase peroxidase enzyme is not produced or is dysfunctional, preventing the activation of isoniazide. Common mutations is S315T, the most prevalent mutation SCR315THR in clinical MDR-TB isolates. Other mutations is GLY316SCR and others in the 2223 base pair gene. Diagnostic relevance is that detection of CATG mutations is critical for diagnosing isoniazide resistant tuberculosis, as these strains are often resistant to other drugs as well. The CATG enzyme itself is vital for the bacteria's survival within the macrophages. While mutations confer resistance, this sometimes result in reduced fitness of the bacteria, although this is less pronounced with the common S315T mutation. So we see here that there are different type of genes whose importance is that they helps in the activation of the antimycobacterial drugs. That is either isoniazide is there, rifampicin, ethambutol. So when such type of mutations occur in these type of genes, it cannot helps in the activation of these drugs, and thus they cannot act on the target bacterium. So basically, these are the some of the important of the diagnostic relevance. The six most frequently reported resistant genes detected are RIF, INH, EMB, OFX, and STR, where RPO, B, CATG, INHA, EMB, B, and GIRA, and RPSL. Drug resistance in the TB complex is mainly conferred through point mutations in specific gene targets through cell wall structure, and the efflux pump also contribute to drug resistance of TB. The reported main amino acid missense mutations associated with drug resistance occur in RPOA, RPOB, and RPOC for rifampicin, CATG, INHA, and AHPC and NDH for isoniazide, EMBB and EMBC for ethambutol, RPSL, RRS, GIDB, EIS, and TLYA for streptomycin, and GYRA and GYRB for fluoroquinolones such as ofloxacin. For example, 96% to 100% of RIF resistant Mycobacterium tubercle isolates have at least one mutation. The most common mutations are in codons 516, 526, and 531 in the RIF resistance determining region of RPOB, which encodes the RNA polymerase subunit. Of INH resistant isolates, 42% to 95% have at least one mutation. The most common mutation is in codon 315 in CATG, which encodes catalase peroxidase, and 8% to 20% have mutation in the INHA open reading frame. Of EMB resistant isolates, 30% to 67% have at least one mutations in EMBB, which encodes arabinosyl transferase. Of STR resistant isolates, 56% to 72% have mutations in RPSL, which encodes ribosomal proteins. Of FQ resistant isolates, 50% to 90% have mutations in GYRA, which encodes the A subunit of DNA gyrase. So here we can, if we summarize here, you can see here that we can see here that this drug was discovered, different drugs was discovered, and what were the genes which were involved in resistance. So here in isoniazide, which was discovered in 1952, and minimum inhibitory concentration was 0.02 to 0.2. The genes involved in resistance is CATG and INHA. And the gene product are the catalase peroxidase enoyl ACP reductase. What is the mechanism of action? Mechanism of action is the inhibition of mycolic acid biosynthesis and other multiple effects. So what happens that there is an inhibition of mycolic acid biosynthesis and other multiple effects. That's why it helps in the it acts on the target bacterium. And mutation frequency is here 50 to 95. Next is the rifampicin. In the year of discovery is 1996. Year of discovery is 1966. MIC 0.05 to 1. Gene involved in resistance is RPOB, and gene product is the beta subunit of RNA polymerase. Mechanism of action is the inhibition of RNA synthesis. Basically, here we are discussing the mechanism of action of the drug, how it act on the drug, and if there is any mutation on these genes, suddenly these drugs cannot act on them. Next is the parazinamide, then ethambutol, streptomycin, then amikacin, kanamycin, capreomycin in the year 1960, quinolones in 1963, ethinomide, PAS, and bedaquiline. Now here, minimum, so these are some of the different type of drugs and their mechanism of actions. So all holds are very important. All these drugs are very important in the diagnosis of Mycobacterium tuberculosis, and any resistance or any mutations in these genes can cause the can help in the non-functioning of these drugs. And because you should be knowing that the mycobacterium drugs are given in combination, and it is a long-term regimen. So it is not a short-term regimen, and if any type of MDR-TB exist in a host or in an individual, it will further aggravate the situation severely, and therefore the treatment becomes quite difficult. So the INHA gene in Mycobacterium tuberculosis encodes an enol ACP reductase enzyme essential for mycofolic acid biosynthesis and cell wall construction. It is a critical target for anti-tuberculosis drugs isoniazid and ethinomide. Mutations, particularly in the promoter region, lead to resistance to these drugs. So already we have discussed all those functions in the tabular form also. So in the last, we can conclude only that it is a major threat. MDR-TB is a major threat to mankind, and it's a major social and economic burden to the society. And some of the important method of treatment is through line probe assay, or we can say ELISA technique, or PCR. Basically, more we are now more emphasis is being laid on the study of the genomes of these drug resistant mycobacterium, so that we can easily identify the target gene which is responsible for the ineffectiveness of these drugs. So we are hopeful that further research and further more work in this field will certainly help more work will certainly help to study this deadly bacteria, and we can certainly limit the growth and spread of this bacteria and save our world from this some of the one of the most of the chronic infections. So with this, I conclude my today's lecture, and I hope you have certainly gained some ideas, or the lecture would have been helpful to you to get an insight into the genomic structure of Mycobacterium tuberculosis as well as the MDR-TB. If you like my lecture, please do like and subscribe my channel, Learn with Ansul, and I value your feedback. Thank you.